I'm the Consul General of Atlanta. I'm really sorry that I'm not able to welcome you in person as we had planned, but unfortunately, the weather does not allow us to do that. As you probably heard, there's a hurricane coming to the coast in Florida, and it will reach the Atlanta region in the night. And as a security measure, many institutions in Atlanta are closed today, including the Georgia Tech campus that was supposed to host the event. I would still like to thank our partner Georgia Tech for co-organizing this event with us. So for the Consulate General of France in Atlanta and for the Office for Science and Technology at the Embassy of France in the United States, today is a big first event. It is the first time that we organize this science summit in Atlanta. It's actually the extension of the annual France Science Summit organized by the Embassy of France in Washington. The objective is really to bring together professors, researchers, entrepreneurs, students from France and from the Southeast of the US to scientific advancements, successful collaboration projects, and also the resources available for our scientific and entrepreneurial communities. This groundbreaking event highlights the vital role of science diplomacy in showcasing the remarkable achievements of French scientists and the added value of bilateral scientific collaboration between France and the US. We are proud that France has long been recognized as a leading scientific nation, contributing significantly to various fields, resulting in innovations that have shaped the modern world. And our collaboration with the US, and especially in the Southeast, has yielded remarkable advancements in research and technology. For example, France and the US are currently co-leaders globally in the number of fields medals, and each of the country has 14 medals as of today, and we were very proud in Atlanta to welcome Hugo Duminil-Copin last year, a French field medal. When it comes to Nobel Prizes across the fields of physics, chemistry, physiology, and medicine, the US ranks first, and France ranks fourth. So these figures eloquently highlight the scientific excellence of both countries. So I hope you will enjoy the rest of the summit, even remotely. And I'd like to invite Bernard Kiplen to share his remarks. Merci beaucoup and have a great event.
Thank you very much, and good morning, bonjour to everyone. So my name is Bernard Kipelen, and I have the privilege and honor of serving as Vice Provost for International Initiatives at Georgia Tech. And it's really a privilege to welcome all of you on behalf of Georgia Tech to this 15th edition of France Atlanta, and especially to that Science Summit. Excuse me, I have a lot of people to thank, starting with Consul General Anne-Laure Desjonquières and her team. Georgia Tech is very proud of that lasting and very productive partnership that has been running for so many years. It's always a pleasure to work together and then to really kind of strengthen the collaboration between France and the Southeast. It's beyond just Atlanta, but really the Southeast of the United States. And as you might have guessed from my accent, I have some French roots. So I'm very, very pleased and very proud that we have that very special partnership. The meeting today would not have been possible without the hard work of many colleagues from the French Consulate at Georgia Tech and at Georgia Tech. You know who you are. The list is very long, so I don't want to forget anyone. So I just thank everybody for their help, and also wanted to thank the the colleagues from our brother and sister institutions in the Georgia Tech innovation and higher education ecosystem. Atlanta is actually a very special place because we are one of the very few cities in the US where we have 3 large so-called research universities. These 3 universities being Georgia Tech, Emory, as well as Georgia State University, but we also have many partnerships with many other institutions, and that makes Atlanta a very vibrant environment for innovation and for education. I want to thank especially Rami, who's no longer with us, who has returned to Paris, but who has really planted the seeds for that event, and also all his colleagues from the French embassy in Washington, D.C. Many of you were at the reception last night, and it was really a pleasure to meeting you. And especially would like to thank Pascal Cohen, his successor, and the new attaché scientifique of the French embassy here at the consulate. That event would not have been possible without its participants, and I know that many of you have traveled overseas, and unfortunately, we have to hold that event online, but we have learned as scientists to be very resilient, and so when it was announced that we had to close the campus, it didn't take too long to basically switch to the online version. And again, thanks to Pauline and Pascal for pulling that together at the last minute. So special thanks to Pauline, I think Claire and Thierry and Florence and others for their hard work in putting this together. I will just end by saying just a few words about Georgia Tech. So Georgia Tech's mission statement is that we are committed to train leaders who advance technology and improve the human condition. I think that second part is very important. We're known to be a large research university. We train our students to advance technology, but nowadays we live in a hyper-connected world, in a global economy. It's very important for our students to also acquire some of these soft skills, and there's no better way of doing that to basically nurture that creativity, adaptability that employers are looking for these days in our graduates than by involving or creating opportunities for these students for international collaborations. And so Georgia Tech is very intentional about its international initiatives, and we're very, very proud of having that very long partnership with the region of Lorraine in the eastern part of France, and where we have an instructional site that was established some 35 years ago, and it's really a transatlantic bridge, not just for education, but also for research and innovation. And I think we have a lot of partnerships with French schools all over Europe, dual degree programs. That also is a way of bringing some of the French students to Atlanta. So we're very, very proud of this. I think that science, transatlantic science is very important, as was mentioned earlier. I think it's really a testimony of building or practicing what we sometimes refer to as knowledge diplomacy. It's very important that we maintain these communication channels. Science is very important in helping us address some of the world's most pressing challenges, and no nation can do that on their own. It really requires international collaboration, and I think it's our president mentioned last night that he attended the Olympic Games in Paris, and he often reminds us that a science like the Olympic Games is an international sport and requires participation from athletes or scientists from all over the world. So I'm so pleased that people are going to share their knowledge with the rest of the group in the following day. And so again, I'm wishing you welcome, but also a very productive day. Thank you very much.
Thank you very much, Anne-Laure and Bernard. And now I would like to mention that as a new attaché for science and technology at the Consulate General of France in Atlanta, It's a great honor for me to welcome you to the first regional edition of the France Science Summit in the framework of the France Atlanta Initiative. First, I would like to express my gratitude to all the stakeholders and collaborators who were involved in the organization of this event. So, as said, it was supposed to be on the Georgia Tech campus, and as pinpointed and highlighted, thank you so much to all the members of the Crisis Board for organizing this event and to make this possible virtually. While I cannot name everyone, I would like to highlight the significant contributions of our partners from Georgia Tech, of course, Dr. Bernard Kipling and his team, and especially Florence Toya. Also, two CNRS researchers, Dr. Claire Berger and Dr. Thierry Lechelet, so thank you so much. All my colleagues at the Consulate General of France, and more especially Pauline Boulet, Deputy Science Attaché for, at the Consulate, and of course my predecessor, Rami Abiakur. I'm also grateful to the dedicated PhD students, Yassine Benzineb and Jan Ferry, and all the others, and Juliette Followay, Deputy Science Attaché at the Embassy of France, who volunteered their time to make this possible. So now let's move on to our scientific program. So maybe we could share the slide. So our exciting program features several high-profile sessions. We will kick off with a fascinating discussion between Professor Raymond Skenazy, whom I will introduce shortly, and Consul General Desjonquières. Following this, we'll have two scientific symposia that will be interspersed by a small coffee break that you could have at home or where you are. These two sessions will bring together French researchers and American researchers from Georgia and Florida to share success stories in transatlantic research collaborations. The first session, just before lunch, will focus on local perspectives from across the southeastern US, featuring professors from universities of Florida, North Carolina, and hopefully Yves Jérôme Baudry could join us from Alabama. After lunch, our fourth session will provide insight into the funding opportunities available to foster French-American collaboration, and we are fortunate to have representatives from various institutions, including the Office for Science and Technology of the French Embassy, CNRS, INSERM, Georgia Tech, and the NFS, the National Science Foundation. Their presentations will be complemented by testimonials from alumni of Franco-American exchange programs. So young researchers were also prominently featured in our program with a poster exhibition, but unfortunately, due to the forecast condition and what we have to deal with in the last 24 hours, it's unfortunately not able to keep that today, but this session will be postponed. Just to provide few information, I would like to inform you that we have received 13 abstracts, 7 from Georgia and 6 from North Carolina, Florida, and Puerto Rico. So we are hoping to organize this poster session maybe next week, and we'll be back to the students, postdoc, and professors who were part of this exhibition. Finally, we will conclude our enriching day with remarks from Dr. Guyarder, Counselor for Science and Technology at the Embassy of France. I would like also to mention that in conjunction with this event, our scientific office was also organizing a meeting with the French tech community in Atlanta tomorrow morning, and we'll be back to you by email to inform you whether this will be still maintained or cancelled. We'll send you an email by the end of the day. I would like also to mention that on September 30, our scientific office will organize with Dr. Anne-Elisabeth Courrier from Emory University an exciting seminar with Dr. Claude Kirchner on the theme of AI and ethics. So this is the overview of our exciting scientific meeting, and even in the conditions we are facing. I wish you all a wonderful summit. So now that the opening ceremonies are complete, let's move on to the keynote speakers session. And it is a great pleasure that I stand before you today to introduce a visionary researcher whose groundbreaking work has profoundly impacted and transformed the field of antiviral therapy. We are honored to welcome Professor Raymond Skenazy. Dr. Skenazy, as you might know, is a world leader in nucleoside chemistry and an expert in drug discovery. His pioneering efforts in establishing one of the first HIV laboratories in the United States paved the way for significant advancement in the treatment of HIV. Notably, he was among the first to advocate for nucleoside analogues as effective anti-HIV treatment. Today, more than 98% of HIV-infected individuals receiving combination therapy rely on at least one of the drugs he developed. Under his leadership, 5 antiviral drugs developed in his laboratory have been approved by the FDA, or more precisely, 26 FDA approvals, including drug combinations, a testament to his innovation and commitment to improving global health. Dr. Skenazy holds over 70 US patents, showcasing his significant contribution to the field. And beyond his academic achievements, Dr. Skenazy is also a successful entrepreneur, having founded several biotech companies, including the prominent Pharmacet, which focuses on innovative therapies for hepatitis C. His relentless pursuit of scientific excellence has not only led to the development of life-saving treatments, but also contributed to the cure for hepatitis C. Dr. Skenazy's remarkable contributions have saved millions of lives, making him a beacon of inspiration for future generations of scientists. Not to mention, he's also a philanthropist, an accomplished Francophile, and a recipient of the Legion of Honor, France's highest distinction with the rank of Knight. Today, we are privileged to hear from Dr. Skenazy in a discussion with Anne-Laure Desjonquières, Consul General of France in Atlanta. And even virtually, please join me in giving a warm welcome to Professor Raymond Skenazy and Consul General Desjonquières.
Thank you, Pascal, for this wonderful introduction.
Thank you. So, Professor Skenazy, I would first of all like to thank you warmly for accepting to participate in this event and share with us your impressive experience and career. Well, Pascal has highlighted how remarkable your career is, and you also have quite an original journey, as you are originally from Egypt. You traveled the world, you studied in England. Eventually, you came here to the US, and you established a world-renowned laboratory at Emory University. You are a friend of our country, France. You speak our language perfectly, and we are very, very happy to count you as one of our most valued friend and partner. So please, could you tell us how your personal and family background influenced your professional choices, shaped your career, and lead you to where you stand now?
Well, thank you very much. This is lovely to be here this morning. As you mentioned, I was born in Egypt, a beautiful country, but unfortunately we had to leave due to the political situation and immigrated to Italy because at the time I was an Italian national, and eventually ended up in a boarding school in England, and from there went to Bath University, studied chemistry, and then went to Yale University, studied for 2.5 years with one of the grandfathers of antiviral agents, Dr. William Prusoff at Yale in the Department of Pharmacology. And that's where a chemist, for the first time, started studying pharmacology and the importance of understanding how drugs disseminate throughout the body and how are they metabolized. And at the time we were working on herpes, there was no AIDS, there was nobody knew anything about HCV. There were many other diseases. We were primarily working on herpes viruses. And today there are 7 different herpes viruses. And from there I wasn't satisfied just being a chemist, so I moved on to Emory and worked in a clinical lab, again, still working on animal models for herpes virus until HIV came along. When HIV came along, we had no idea whether it's going to be a fungus, a bacteria, or some other weird microorganism. It turned out to be a virus, and I was perfectly trained to work on these viruses, having worked in Bill Prusoff's lab and with my knowledge of chemistry. So I started making molecules and set up the first HIV lab at Emory University. And many people wouldn't take that risk. I took that risk. And I think the results became very obvious after a couple of years when we discovered some early, not so successful antiviral agents, but then later on, some of the most successful antiviral agent on the planet, specifically emtricitabine, lamivudine, and then other anti-HIV compounds that became, but they're routinely used all over the world. So I think this is sort of the journey that we took from Egypt all the way to Emory. And, you know, it's a matter of being prepared, taking risks, and knowledge. Knowledge is incredible because we knew what the target could be for HIV very early on, and we had started to make molecules that could work against these targets specifically without affecting normal cells, and that's what made these compounds very valuable.
Sorry. So thank you for this. As you mentioned, you're really a leader in antiviral drug discovery. Your lab has advanced several FDA-approved antiviral agents. You explained us that you were able to take risks, and this was one of the key of your success. Your career encompasses numerous patents, high-impact publications, and also a very successful entrepreneurial journey. Could you please elaborate a bit more about this aspect of your work, and also maybe tell us, among your so many achievements, which ones you are the most proud of and why?
You know, success has many fathers, as we say. And it's not just me. There was a whole team of people behind me to develop these molecules. And the frustration for scientists, even scientists at Georgia Tech, is they make discoveries and nobody believes them. And you really need to be a champion to take these molecules forward and your ideas forward and convince the world that you have something useful. Also, the funding is important too, because if you don't have the funding, you can't do anything. And fortunately, I was funded by NIH, and initially not, but later on, yes. And the money comes through, and then we can do a lot of things. And that was critical. And the important thing now is that these molecules not only are developed in a university setting, but then you need to find a partner, either form a company yourself or find a partner, license the technology to a company, whether it's an international company or a small company, or start up your company. Unfortunately, nobody was interested in my drugs at the time. So I had to form a company. And it's not easy to form companies. I mean, I'm not set up, I'm not trained as a businessman initially. And I had to basically start looking at funding, potential funding, to take these drugs further than just a university environment where we can take drugs all the way to a lot of preclinical studies, but we can't do really the clinical studies because of the cost and also the API, the material that you need to make the drugs. is very large commitment. So we had to form the companies, and then of course the companies developed these drugs as fast as they could with all the hurdles. Now, when you have a good molecule, fortunately my molecules were good ones, and we had failures, of course, but the ones that were successful, every experiment you do actually worked because it's a good molecule. And that makes things a lot easier. And you have little toxicity or no toxicity whatsoever, because that's the main reason why drugs fail, is due to toxicity, not usually due to inactivity. And that's really quite important. Now, one of the two most successful drugs I think we've made and we've been involved with are the drugs for HIV, a class of compound which are nucleosides. They're building blocks for DNA and RNA. And these molecules are very special. They're unique. They've never been made before. They're not repurposed drugs. They're not drugs you take up from the shelf. You have to actually design them. The same way as an artist design a painting and thinks of a painting in the same way as we saw yesterday, Plantier draw something creatively. We also do the same thing. And it's a bit of trial and error, but it's also based on all the education you've had and all the knowledge you've accumulated over decades. And you can build these molecules and make them synthetically in your lab and then test them in robust assays. And that's what makes these molecules successful. So to talk about the two molecules briefly, one is an HIV drug. And what it is, it's a new concept. It's a completely new concept at the time, because most nucleosides in nature are of a particular conformation, a particular shape. And it's like your left hand and your right hand. And most of the nucleosides in your DNA are basically right-handed. And we made the mirror image of this molecule, completely different. That's what's expected and not usually found in nature. So by making these molecules, we fooled the virus into taking these molecules and basically incorporate them into their DNA. And when you do that, you cause a chain termination. The DNA doesn't elongate anymore. And that's how you create this molecule that's been so successful and so effective. And it only does it with a viral enzyme. It doesn't do it with a cellular enzyme. That's where you get the specificity. In terms of the second drug, sofosbuvir, this is an amazing story. I won't have too much time to talk about it, but basically, again, it's a novel ribonucleoside with a fluorine in a certain position that's quite important. And the molecule itself is not active. But when you make what we call a prodrug, the cells take up the prodrug specifically in the liver, where the virus is. The liver hepatitis is in the liver primarily, and it degrades the prodrug into the drug itself, which is then activated to the active metabolite that inhibits the virus. So it's a very unique concept of a compound that's totally inactive. And that's one of the reasons why it's such a safe drug, because it's inactive normally unless you make the prodrug, and then it goes to the intracellularly into the liver and gets degraded and metabolized in the liver, releasing the active moiety. And that's why it's such an effective drug. And it's not just any drug, it's a cure for a viral disease. It's the first cure for a viral disease on the planet. And it's never been done before. And that's why it's such a huge success, with today more than 13 million people cured of hepatitis C thanks to this drug.
That's really, really very fascinating. Thank you for this. We've spoken about the good molecules, the successful ones, and they were very successful. But you also said once with humility and irony that you have 4 cabinets full of unsuccessful grant applications. We all know that the world of scientific research is very challenging and competitive. So what message could you give to our young PhD students and researchers regarding resilience in their careers?
Well, first of all, you have to have good mentors. I think it's important, and then you have to put in the hours. You can't just not be in the lab trying out things. And then if you have an idea and you think it's a good idea, start by proving it that's a bad idea. And then if you're successful, then you've proven yourself that it is indeed a good idea. So it's known in science, the null hypothesis. So you start with a negative thing, and then you turn it from negative into a positive thing. But I think the most important thing is persistence, as mentioned. Being persistent in your ideas is so important when you're young. And you need to talk to some of your mentors. That is so important. And they will be the ones who will guide you, hopefully guide you. But don't always listen to them. That's my advice. Don't always listen to them, because if they say no, go into the lab and prove them wrong. And if you can prove them wrong, you will have tremendous success, and you'll have more confidence in yourself as you move forward.
So techniques in research are evolving, and could you maybe tell us what you think about the role of in silico and computational approaches in antiviral drug discovery, and how to predict drug resistance mutations in viruses? Maybe can you also tell us if you see artificial intelligence having a place in these strategies in the future?
Yes, I think AI is becoming more important now. It's not. We've been using AI for a long time. It may have been called something else, but one good thing about chemistry is that we've had computerized systems for a long, long time. And in fact, some people said we don't need any more organic chemists. The computers can do it all. Well, that's not quite true. You still need the brain, a human brain, to function, and I don't think we're finished with that. So, and you have the wisdom, because it's something else that comes into play, and that's pharmacology, as I mentioned earlier. You can make the computer can design drugs with your help, but then you have to also work out the pharmacology and the safety of the drug to make it a successful molecule. So that will take a lot more effort, and people are working on additional AI to learn about how these drugs are metabolized. And in fact, once you learn how it's metabolized, you can go back to the lab and make analogs that will be less susceptible to metabolism, and using the knowledge that you gain from the AI information that you receive. So there's a multitude of ways of doing things, but I don't believe in doing things randomly. You have to do it. You have to do it with the knowledge and the wisdom that you've accumulated over the years. Plus AI, you're in a very powerful situation. And in terms of resistance is, we say, is a sine qua non of antiviral agents. And usually you can always select for resistant viruses in the presence of drugs. That happens also in nature. The selectivity, the resistance emerging in nature and emerging in patients, and that could be devastating. The ideal drug is the one that has a high barrier to resistance. So again, you can develop drugs that have a very high barrier, and you can look at the structure of the enzyme. You can now go back to the lab and design the molecule so it has this high barrier, and that can help. But in addition, we've used combination chemotherapy, making it a lot harder for the virus to select for resistant virus. Even for some fabulous drugs, it's important, like sofosbuvir, the drug that cures hepatitis C virus. We still need a second drug to prevent resistance from occurring and ensuring that there is complete suppression of the virus and eventual elimination of the virus.
So still looking towards the future, and we have new viruses emerging all the time. Many viruses also that have been there for a long time, and that we still do not know how to fight against. Could you tell us, what do you see as the biggest challenges ahead in antiviral research and innovative vaccines, and maybe share how optimistic you are about the future and about the way we are going to be able to overcome these challenges?
Well, it's partly the emerging viruses are really, especially the ones that are mosquito-borne, we think that with global warming, this is going to cause more viruses migrating up north from Brazil, from Venezuela, from Colombia, from Puerto Rico into the United States and other parts of the world. Or in the case of France, the Mediterranean, we're starting to see diseases like dengue and West Nile, which are flaviviruses. These are important, the same class of viruses as hepatitis C virus. And we're seeing more and more of that. We don't have any treatments that are really effective against dengue and West Nile. And there are many other viruses that you probably never heard of. Chikungunya, for example, there is Mayaro. I can keep naming a whole bunch of viruses that are potentially emerging with time. They will become more familiar, a bit like nobody knew what coronaviruses were. Now everybody knows what coronavirus is today. Things change, but we do have significant problems with tick-borne viruses in many parts of the world. We have West Nile is a serious problem affecting over 1 million people in the United States. We heard recently that Tony Fauci had West Nile virus disease. So they're painful arthritic type of diseases that have lasting consequences, and there's no treatment available. There's no vaccine either. So vaccine goes hand in hand, and prevention is certainly very important. But when you fail with it, we don't have a vaccine, you have to develop drugs or have a drug as a backup just in case. So we're going to see that. And I think one of the viruses, if I were to guess what could be the next big plague, it could be other than avian flu, which is currently a big problem, beginning to be a big problem in the United States. I think monkeypox is certainly one to think about, especially since it's still brewing in Africa, and you're now seeing it in parts of Europe. And we had also a strong bout of monkeypox in the United States, but fortunately, measures were taken to prevent the spread of this virus, which can be quite devastating. It's a virus, of course, related to smallpox, which has been eradicated. And one of the biggest successes in the history of mankind was the elimination of smallpox. Hopefully, we can eliminate monkeypox as well in the future with vaccines and also drugs.
Well, thank you so much, Dr. Shinazi. I think we are running out of our time. That was very enlightening, and we can only hope that against all those emerging viruses, there will be many researchers as successful as you were and as you still are. So thank you very, very much for sharing your experience. And we will continue with the further discussions of the summit. Thank you.
Thank you very much. And we are continuing training the new generation of virologists, and we need more virologists in this world because there's going to be many more problems. Thank you very much.
Thank you, Professor Shinazi and Consul General Dejonckheere, for that insightful discussion.